So the buildup for technology is finally here. No pressure, Rhodes. But our next speaker is the founder of Trusell's Technologies, Dr. Rhodes Trusell. It's an environmental engineering firm focused on water treatment processes and water quality. Rhodes received the NWRI Athalie Richardson Irvine Clarke Prize in 2013 in recognition for his 40-year career focusing on improving designs of new water treatment plants and technologies. He has a special interest in emerging water quality problems and reuse. Because of his demonstrated expertise in these fields, he's been invited to serve in numerous leadership positions, including chair of the Water Science and Technology Board for the National Academies, chair of the Research Advisory Committee for the Water Reuse Research Foundation, chair of the Editorial Advisory Board for Standard Methods for the Examination of Water and Wastewater. He was also active in US EPA's Science Advisory Board for 17 years, served as chair of the Committee on Drinking Water as well. Rhodes has his BS, MS, and PhD in environmental engineering from a place called UC Berkeley. So with no further ado, Rhodes Trusell.
Well, good morning. There's been a lot of presentations on the Drug Potable Reuse Initiative and its research program, and so I'm not just going to summarize that. I instead, I hear it ringing. I hope that's
just your ears.
Not going to get worse. Anyway, I don't want to just summarize that program. What I'm going to try and do is offer a way of looking at the program and perhaps making some decisions about investments there. So with that, I want to give acknowledgements to two gentlemen who give me a lot of feedback as I've generated this, Shane Trusell and Brian Pexon. This slide here is a slide that Doug Owen used about a year ago during the opening meeting with the panel that we had on that we have for the state of direct potable reuse to summarize the research that's being done to support the direct potable reuse initiative. And it begins all the way on the upper left there with that workshop in Sacramento in April 2020, and then if you go 2010, if you go all the way to the right on the lower orange, that box is the panel's presentation in 2016 of their assessment of the feasibility of direct potable reuse and all the activities that have gone and will go on in between. That chart does not have one activity that took place just last week, which is a state board workshop on recycled water research needs held in Costa Mesa here, but I want to use that as a place to start because it's the most recent discussion on the topic. So the workshop looked at 4 thematic areas: water quality and human health, performance reliability, ambient water effects, financial, environmental, and social factors. I was not at the workshop. I had the opportunity to review a short write-up on each of these thematic topics, and each of them identified research needs. And the group then went on to discuss those research needs, and I'm not privy to the outcome of that discussion. I've not heard any summaries of it yet. Not that they won't be available, just Friday?
Right, it'll be 2 weeks.
So, but I do want to offer a structure which we thought would be useful for looking at the kinds of decisions we make about investments in potable reuse. So on the y-axis on the left is the length of time required to achieve the research objective, ranging from, short term, you can leave it down there for now, to the long term. And then the x-axis is a qualitative measure of the investment required. So on the left you have, local funding, and on the right you have much more massive funding like we would have to have from the federal government, or perhaps even more than the federal government could afford. Both these scales, I think, should be viewed as logarithmic. So on the upper right, We have objectives that will take decades of time and truckloads of money. An example that comes to mind for me is cold fusion. So here I have laid out some of the research needs that were presented in those write-ups. And you'll note, and I don't tend to go through all those, you'll note that basically those that take more time also take more money. No surprise there. I want to focus just on those on some of the extremes, especially the ones on the upper right. And one of those is continuous monitoring of pathogens in drinking water. And another one, and to make sort of parallel to that need, I'm proposing to combine three others. So a cancer screening method, human health relevance of bioassays, and a CEC Risk Paradigm into another title, which I'll call Continuous Monitoring of Chemical Risk in Drinking Water. So I have these way on the upper right, should they be there. So let me talk about that a little bit. So you remember last year I had this example where I tried to compare NDMA as a chemical and I believe I used norovirus as a pathogen. This time I'm going to do it with rotavirus for various reasons. Same exercise, but I don't want to go through all of them. I'm going to take a different look at it this time. This time, what I want to do is talk about from the perspective of these two contaminants, what would have to be done to monitor each of these contaminants directly and demonstrate that they are now at safe levels. Okay, well, let's first look at NDMA. Here in California, our arbiter on these issues, OEHA, has studied this chemical and they've proposed a public health goal, which essentially is a safe level of 3 nanograms per liter. Using NDMA as molecular weight and Avogadro's number, you know, I'm into that kind of stuff. I figure we would need a probe that could demonstrate that the sample had less than 10 to the 13th molecules per liter. Okay, now let's look at rotavirus. In a paper in 1991, EPA looked at rotavirus and they proposed a goal which corresponded to a risk of 10,000, one infection in 10,000 people per year. And that level they estimated using quantitative microbial risk assessment was 2 times 10 to the minus 7 viruses per liter. Now, it may seem like a ridiculous number. People have told me it is, but keep in mind they're looking at these 10,000 people collectively drinking 7 million liters of water during the course of that year, and they don't want any of them, they only want one infection. So here's kind of a summary of where I ended up there. If we assume a perfect technology, so that is, if there's only one virus in the sample, we'll find it. If there's only one molecule of NDMA as well, we'll find that. I mean, I don't want to have any limitations on technology here. Then we would have to sample on the left there for the viruses 10 million liters of water with this probe. And in the case of the NDMA, we'd have to sample 1/10 of a picoliter with our perfect NDMA probe. So what kind of sample volume do our current analytical methods look at? Well, for viruses, we typically use cell culture or PCA. There are some other methods out there, but these, I think, are the more sensitive methods. Both methods process around 1 milliliter as a sample, give or take. For NDMA last year, Shane Snyder did a great presentation promoting a new, Shane Snyder's our guru in this area. Another one is our prize winner this year, but I don't think they differ on this a lot. But Shane was promoting a particular new method he had with MSMS, where injecting only 50 microliters in his machine, he could get a detection of 10 nanograms per liter. So that's within a factor of 3. We're almost there, right? So 50 nanoliters, or 50 microliters. Now, to be fair to the viruses, we can concentrate. We can also do this with the organics, but we don't have to. So let's look at the microbes. We can concentrate about 100 liters with some of the methods we have out there and improve our situation. Of course, the concentration methods don't get real good recovery, but let's, for the moment, dismiss that issue as well. So we have 100 liters. So now let's look at what that gets us. Let's first look on the right. Our perfect method that detects only one molecule would need 1/10 of a picoliter. And one of the more sensitive methods we have out there already can look at 50 microliters, which is a much larger volume. And its detection was already in a factor of 3. So I would say, looking at that, I think we can conclude that online analysis to demonstrate NDMA is at a safe level may be achievable. I'm not trying to say we're here today, but I mean, it's not ridiculous. Now let's look at the viruses. Our perfect method that detects one virus would need to survey a 10 million liter sample. We have 2 methods. They can both handle around 1 milliliter, but with concentration of 100 liters. That puts us between 10 to the 5th, well, between 5 and 10 orders of magnitude difference between our sampling method and our goal. So I come to a completely different conclusion here, which is online analysis to demonstrate that viruses are at safe levels is not achievable. Well, in my lecture last year, I said the science of water analysis is the fastest growing area of water technology. Remember that chart showing the detection limits are declining like, what's the law in electronics? Moore's Law. And again, I'll mention Shane. Shane is one of our major advocates of all this new technology. Shane has gone around, Shane Snyder, and given wonderful talks on these new technologies. So I'll take it from there. At the leading edge of this technology, one of the leading edges, is biosensors. This is a marriage between biological sciences and the world of the electronic microchip. This is an exciting new technology based on sensors that transform biomolecular reactions like enzyme reactions or antibody reactions or even the metabolism of a microorganism into an electrical signal. They really are probes. I mean, they're amazing and they're real. Let me give you an example. This thing here. This is a glucose monitoring thing for diabetics. I use it several times every day. This thing needs a sample of 4 microliters, which is really great for my fingers. Less than 1/10 the sample that Shane needs in his MSMS. And it's all I need. It fits in my pocket. That same technology is now being used to make artificial pancreas for diabetics. This is not tomorrow. This is today. You can go buy this thing from Medtronic. You can even get Medicare to help you pay for it. Type 1 diabetics, not type 2 like me. Another concept is the lab on a chip. It integrates several laboratory functions into a single chip. The whole thing is just a few milliliters in size. I mean, lab on a chip has its own journal. It has a wiki site. I mean, if you have your own wiki site, you're there, right? So can't all these wonderful new technologies solve our problem? Certainly they can get the answer faster than our conventional techniques. I mean, this little thing does my blood sugar in seconds, 10 seconds to be exact. And not only could they tell us the virus is there, they could tell us about its parents and grandparents, right? Unfortunately, one of the greatest strengths of these technologies is also their weakness. My glucose monitor, for example, analyzes, I told you, it analyzes a 4 microliter sample, not 100 liters or 100 million liters, but 4 microliters. So, the problem is there's just so much water. Or put another way, finding one virus in 10 million liters is worse than finding a needle in a haystack. I mean, the problem isn't identifying the needle and its grandparents when you find it. The problem is there's so damn much hay. So what strategy can we follow? We can do what we do already in drinking water today, but because the pathogen threat is higher, I'm still worried about the pathogen threat. Particularly if we're going to do potable reuse, we should probably do it better. So, what are we going to do? We're going to decide how much removal we need to accomplish it. We're going to identify the critical process for doing it. We're going to design that process for reliability, and we're going to monitor it. So, I've concluded that continuous monitoring of pathogens in the product water to prove it's safe isn't viable. Do we also conclude that monitoring chemicals in drinking water is easy? Well, certainly we can conclude, I think, I can't see my slides here. No, I'm going to stay on this one. Certainly we can conclude that the monitoring of chemicals in drinking water is possible. But monitoring chemical risk is not just a question of monitoring the chemicals, is it? I mean, remember my chemical risk concept, what I was doing was using an umbrella to try and capture cancer screening and, health relevance of bioassays and the CEC Risk Paradigm, a lot of things that people would like to see. Let's try and look at each of these in a little more detail. So first, we have the cancer screening risk. In the early 80s, NRC developed a protocol. I have to go back. For me, this history is so important. So in the early 80s, the NRC developed a protocol for testing concentrates of organics in drinking water in connection with a proposal to do potable reuse in Washington, D.C. They didn't want to build dams, so they should do potable reuse. Obviously, they didn't do it. So they recommended that we do in situ mutagen Ames assays, the Ames assay that was developed at UC Berkeley. And at that time, there also had been a mammalian cell transformation test that was more indicative of cancer that had been developed at USC, so they recommended those 2 in situ tests. Then they also recommended that, and these would be done concentrates of the organics, and they also recommend these concentrates be applied to in feeding tests, animal feeding tests. And the experimental estuary water treatment plant, which was a large full-scale study that was done to look at potable reuse, Mike Kavanaugh led this research project, did both the in situ tests, and they found similar results for the recycled water and all the local waters. But in reviewing it, the NRC said that they really need to see the whole animal feeding test to look at it. They said a lot more, but that was one of the requirements. Whoops. So projects in Tampa and Denver subsequently did do the whole animal feeding studies, and they showed no adverse effects. Then in 1998, another NRC committee I was on, I didn't write this part of it, but found that data was too limited to draw any general conclusions. Basically, what we concluded was those tests did not show evidence of a problem in the water that was tested. Keep in mind, these are millions of dollars worth of tests, but that didn't show that the water was safe the rest of the time, and for that matter, it only showed no evidence. It didn't show proof of no risk. Also, you should be aware, some of you are, but some of you are too young, the federal government has been searching for a valid cancer screening test ever since the Delaney Amendment was first passed in the 50s. It's been something repealed, but that effort begun then, and after hundreds of millions of dollars and decades of research, I actually think the scientific community as a whole is less confident in our ability to do that today than they were in 1980. So as I consider this history, I'm inclined to move our screening test to the upper right. So what about the CEC Risk Paradigm? You got a lot of insight from that already, I think, from the discussion that Rhodes held earlier. This is another question we've been looking at for a while, not as long. Research in the mid-90s got us concerned about hormonally active agents in the environment. At the turn of the century, with NRC's help, EPA began to look at how we could design a candidate contaminant list for our drinking water that would give a way to take a broader look at all these compounds that we're inventing all the time and how they could be assessed. I was on that committee. And at the same time, we began to find pharmaceuticals and personal care products in our lakes and rivers and water supplies, and indeed in some of our drinking waters themselves, as Urs mentioned earlier. And WHO and EPA and the Australians, Urs mentioned one of their efforts, and various institutions throughout the world, including even here in California, the state board, all struggled with what would guidelines look like, how would we manage this issue. So where do I think we stand now? Well, I think there seems to be general agreement among scientists and public health experts that the risks with the CECs aren't great. That doesn't mean that there's agreement they should stay there, but I think there's general agreement that the risks are not real great. But there's no public consensus on this matter at all, and none that I can see coming soon. So my view is the CEC issue will continue to be a challenge for some time, but it's probably not way out there like cold fusion. So the remaining issue is the relevance of bioassays. Now, I haven't had the opportunity to review all the work done on that today. There's one report by the Water Reuse Foundation by Beate in Australia that I've seen. There's other work that's been done by Squirt that I haven't had. It's much more, almost 10 times the investment. I haven't had the opportunity to look at that. I don't think there's a report yet. But I think it's clear that bioassays are making and will continue to make, and I also Urs did some of that in his talk, important contributions to our understanding of chemicals, the environment, and they're going to be useful tools for evaluating environmental impacts and probably also useful for looking at drinking water sources as well. But I'm reminded again of that statement the NRC made in that 2012 report, which is unlikely that any laboratory test will ever establish the absence of health risk from a drinking water source. So I think as we think about developing and using this tool, I put it another way, bioassays may be an important step in improving our understanding of water quality and health, but we will not be able to use it to prove the absence of risk in conventional waters or our potable use supplies. So I don't know where it belongs, but I kind of put that in the middle somewhere too. I think the reasons for considering potable use are pretty compelling. They have to do with the risks that I think, I read David's paper, that I think David will talk about this evening in his lecture, and the fact that potable use along with water conservation, which was just mentioned earlier, have the potential to get our urban communities through the bulk of this century, I think. But if we're going to do this safely and timely, I think we're going to have to make investments in the issues that I've circled on the red on the left. Questions of finding and demonstrating surrogates and indicators, demonstrating reliable systems of real, potable use systems. So, in summary, our potable use agendas continue to develop. Some important questions are being raised, but some of them are deeper than the potable use issue alone. I'm reminded of our friend Donald Rumsfeld, who was much maligned for what I actually think was one of the most intelligent statements he ever made. As we know, now let me just repeat, as we know, there are known unknowns. There are things we know. And we also know there are known unknowns. That is to say, there are some things we do not know. But there are also unknown unknowns, ones we don't even know we don't know. Isn't that great stuff? It really got him into a lot of trouble, but I think it's a great insight. And we have to be careful not to burden our effort to do the research necessary to make potable use safe with the burden of solving these conundrums we've been struggling with for decades. Thank you very much.